Welcome to the video about antibodies. I mentioned in the introduction to the immune system that really the function or the purpose of the immune network is to defend the body against danger, and that a lot of times that danger is in the form of infections with viruses, bacteria, parasites, organisms that are oftentimes microscopic and oftentimes are called pathogenic. And that's a fancy word, so let's write that down. Patho means disease, and genic means kind of giving rise to or giving birth to. So pathogenic microorganisms are ones that can cause disease. Now, the distinction between pathogenic microorganisms and bacteria or viruses that are not pathogenic is sometimes a little bit blurry. There are, for example, bacteria that live on our skin or in our intestines that are actually helpful to us as long as they stay in those anatomic locations and kind of behave themselves. In fact, they can be quite helpful. But if they gain access to a tissue where they don't belong, say deeper structures in the skin or in the bloodstream, then they can cause disease, and they are then classified as pathogenic. So how does the immune system actually defend the body against danger when, let's say, a pathogenic microorganism like a bacterium enters into tissues and starts causing damage? How does the immune system respond? What mechanisms are available to it to actually stop the invasion? So antibodies are one of the mechanisms that the immune system has. And in this discussion, I want to remind you that the immune system really is a processing system for stuff, for matter. And on the molecular scale, it basically works by shape. So the immune system is sensitive to molecular shape. And it basically recognizes danger by, not shaper, just shape. It recognizes something dangerous by essentially feeling the molecular structure. And depending on context, it'll decide whether that shaped molecule is dangerous or not. So let's say that there's some evil bacterium. Here's an evil bacterium. And it has a nefarious growl and some teeth or fangs here. Of course, it doesn't really have that, but it suggests they got kind of poetic license here. And it has some proteins on its surface. And let's say that this evil bacterium gains access to the deeper layers of, let's say, the skin. There's, let's say, here's the skin, and the skin is all good, but then there's a break in the skin, and through this crack in the skin, or fissure, this bacterium starts to enter into tissues where it doesn't belong. So it kind of enters into here, and now here it is, and unfortunately it's all happy because it has gained access into the deeper tissues, and there it finds lots of nutrients, and it starts to cause damage to the cells of the lower layers of the skin, for example. And this could be true of almost any tissue, but let's say the skin for now. So one of the ways that the immune system has of combating this invasion is by producing a certain class of proteins, as I mentioned, called antibodies, and I'll abbreviate antibodies here as AB. Antibodies are also sometimes called immunoglobulins, and that is a super fancy term, immunoglobulin, or sometimes that's even abbreviated IG, and that'll be important for looking at what are called the subtypes of antibodies or the classes of antibodies. But immunoglobulin just means immune protein. Globulin means protein, and immuno, of course, means immune system. So these are specific immune system proteins. So how does this work? Well, I'm going to draw these in a kind of cartoon form, and this is a common form that you'll see. There's a long tail to these proteins, and then they have kind of two arms, and at the end of the arms are these kind of claws. And remember, the immune system works by shape, so I like to think of the immune system's antibodies as kind of molecular attack dogs. They have a long tail, then they have two arms, and then the claws here at the end are the critical pieces for the recognition of a specific invader. So imagine that you had an attack dog trained to guard your yard. And let's say that some evil terrorist was prowling around the neighborhood, and your dog was trained specifically to recognize this terrorist. In fact, the claws on your dog's paws were just exactly the right shape to bind onto and grasp onto the leg of this dangerous terrorist that's roaming around your neighborhood. Well, your dog roams around your yard, and if that terrorist enters your yard, that's great. This dog is all ready and clamps right onto the leg of this invader. And when those claws clamp on, they do a couple things. First of all, the claws kind of dig into the surface of the invader here, and that knocks holes into the invader. Secondly, it kind of impairs the function of some of these proteins on the surface that help the bacterial invader continue its invasion, and so that frustrates and slows down the invader. And then, last but not least, this tail here can act like a sort of handle and a cell of the immune system, kind of like a police officer, can grab onto that tail, and using it as a handle can kind of wrap around and engulf this bacterial invader and actually digest and destroy it. So here's, the nucleus of a cell of the immune system. This might be a macrophage or a dendritic cell. That name isn't really important right now, but what is important is to see that a lot of this inhibition of or blocking of this invasion is due to the antibody. Okay, so here's the problem. Let's say that a second terrorist enters your neighborhood and sneaks into your yard. Well, the antibody against the first invader, with its claws that are so specific for the first terrorist, is of absolutely no use in combating that second terrorist. Why? Because the second terrorist has a completely different shaped leg. It has different proteins on its surface. It's a different organism. And because the immune system works by precise molecular shape, and these claws are so precise, that first antibody is of no use against the second invader. So that means you have to have a second antibody attack dog for combating the second invader. And in fact, because the outside world, the universe at large, is so full of millions, billions, or perhaps trillions of different organisms with slight variations sometimes, even closely related bacteria or viruses might have slightly different structures on their surface. The immune system is very sensitive to those variations and, in fact, has to generate a new antibody and kind of train up a new attack dog to address each and every one of those invaders. So you don't have one or two dogs roaming around your yard. You have hundreds of thousands or even millions of different antibody dogs all roaming around the yard of your body, so to speak, each one looking for its particular target. And in fact, you have multiple copies, hundreds of thousands or millions of copies of each one of these. And I'll just take a brief aside to say that it's largely the goal of immunization to produce antibodies. In other words, what are you doing when you get the annual flu vaccine? So a vaccine, of course, is a dead or weakened version of an invader, and what you do is you intentionally introduce it into the body in a weakened or dead state so that the immune system cells grab onto it, maybe even with a weak antibody or without any antibody whatsoever, and then the immune system processes it and produces these antibody dogs so that the immune system is kind of like on high alert. And if the full strength invader should come along, not from a vaccine, but from, so to speak, natural infection, then you've already got the antibody dogs waiting to surround and engulf this invader and destroy it. So I see that I'm out of time. In the next video, we'll talk about the subtypes of antibodies and what they do. See you in the next video.